Hi everyone, my name is Love and I'm one of the SIs for Dr. Maduro's BIO005A class. And today I'm going to be going over the first half of one of Dr. Maduro's practice midterms. And to jump right in, number one starts off by saying the diagram on the right is a mitochondria, and it's going under cell respiration in the presence of oxygen. And it's saying that structures 1, 3, and 5 are regions of the cell and the mitochondria, while 2 and 4 indicate membranes. All right. So it's saying which of the following are correct. So I'll go in order from A through E, and we'll determine which one's right. So A is saying glycolysis occurs in region 5. Well, region 5, which is right here, is saying it's in the mitochondrial matrix, right? Well, we know glycolysis doesn't occur in the mitochondria. It occurs in the cytosol. It occurs in region 1, right? So we know that A is most likely not true. B, the electron transfer chain is found in region 4. Well, electron transfer chain occurs in between the matrix and the intermembrane space, right? So it occurs in the inner membrane. And so that is region 2, right? Region 4 is the outer membrane. So B is not right. C, the lowest pH is found in region 3. So as we said in the electron transfer chain, the protons are pumped from the matrix out to the intermembrane space, right? So if you pump a bunch of protons into one area, it's going to increase acidity, right? Which is defined by a low pH. So yeah, that makes sense, right? Since region 3 is the intermembrane space where you pump out all your protons, it's going to be more acidic, thus you'll have a lower pH. So let's keep C for now. D, the citric acid cycle occurs in region 1. So region 1 is the cytosol, and we know that's where glycolysis occurs. Citric acid cycle is in the mitochondria, so that would not work. And finally, fermentation makes carbon dioxide at region 2. Fermentation also takes place in the cytosol, so not in the mitochondria. So E doesn't make sense either. So yeah, our final answer for this question will be C. All right. So number 2 is saying during the reactions of the citric acid cycle, basically what happens, right? So let's go through what happens in the citric acid cycle first. So we know that it goes glycolysis to pyruvate, so I'm gonna write G, goes to pyruvate oxidation, right? So PO, and then you go to the citric acid cycle, right? So let's see. So what enters the citric acid cycle? Well, you break down glucose into pyruvate, and then you take pyruvate to acetyl-CoA. So that's what enters the citric acid cycle, right? So acetyl coenzyme A enters the cycle, and let's just talk about what gets released in this cycle, right? I apologize for my drawing in advance; it's really bad. But well, we know that we get 2 CO2s, right? 2 carbon dioxides. We also know that we get an ATP molecule. We also get a lot of NADH, 3 of them. And we get another carrier, electron carrier called FADH2, right? So yeah, so these are so we put in acetyl-CoA and we come out with all these products, right? So let's go through what the answer choices say. A, the electron carrier FADH2 will undergo oxidation. Well, oxidation is technically the removal of the hydrogens. So you can think of anytime you're adding hydrogens as reduction in this class. So if you're making FADH2, you're actually going from FAD plus to FADH2, right? So since you're adding some hydrogens, you're reducing it, right? Not oxidizing it. So that's not right. B is saying acetyl-CoA is regenerated every turn of the cycle. Well, acetyl-CoA, it's going to be hard to regenerate it, right? Because it's a 2-carbon chain that comes from the breakdown of glucose. And if you're breaking it down into carbon dioxide, it's going to be hard to regenerate more carbons, right? Essentially. So that's not going to work in this case. 2, there is an energy investment phase requiring ATP. So that's not true because we know that it actually releases ATP down here. C or D is saying most of the energy in glucose is used to make ATP. Well, we know, yeah, energy or glucose does make ATP, but most of the energy, I would say, is probably going to be made in the oxidative phosphorylation stage. So that doesn't really sound right either. And finally, high energy electrons are used to reduce NAD+. Yeah, so we know that NAD+ is an electron carrier, right? And the way it carries electrons is when it gets reduced to NADH. So that definitely makes sense, and that's definitely true. So E is our answer in this case. So let's go on to number 3. Number 3 is asking the electron transport chain. So I'm just going to flash a picture of the electron transport chain on the screen real quick, and it'll help us review what we're talking about. So it's saying the electron transfer chain allows protons to diffuse across the mitochondrial membrane. Well, it's technically not diffusion because the protons are pumped out of the matrix into the intermembrane space using the energy that comes from this coupled protein carrier transfer of the electrons from NADH, right? So it's technically using some sort of energy, right? So definitely not diffusion. And also, if you think about it, you're going from a high concentration to a low concentration. I mean, you're going from a low concentration to a high concentration, right? So since you're going against a gradient, you're going to have to use some sort of energy, right? Let's see. B, reduces oxygen using electrons that are stored in ATP. Well, ATP doesn't really store electrons that can be used for energy. The energy is stored in the phosphate bond. So this doesn't really have any correlation to do with the electron transfer chain. So B is not right either. C, converts energy from NADH and FADH into ATP. Well, that's kind of like the whole process of oxidative phosphorylation, where you're using NADH and FADH to make ATP, but the electron transfer chain does not directly do that, right? You have to go through a series of steps before you make ATP, right? It's not direct. So C is not right. And D, we move electrons from the matrix to the intermembrane space. So this is a big confusion that a lot of people have. We're not moving electrons, we're moving protons, right? These plus charges are the hydrogen, so they're protons. We're moving the electron through this right here, a series of complexes in the inner membrane, all right? That gives us energy to pump out these protons to the intermembrane space, all right? So D is technically not right. And E says uses energy from electron carriers to pump protons. Yeah, so we're using these energies from these electron carriers, right? Because they hold our electrons. And since they're moving through these complexes, they're producing an electrogradient that allows these protons to be going against their gradient into the intermembrane space. So E is definitely our answer. All right, so 4 is asking, when fats are broken down into our cells, glycerol is generated. It can be modified to produce a phosphorylated 3-carbon sugar, G3P. Then the G3P could enter cellular respiration during the steps of. So it's basically saying that we have a 3-carbon sugar, and it's asking what part of the cellular respiration cycle can this 3-carbon sugar enter, right? Well, we know that basically that glycolysis is breaking down of a 6-carbon sugar, right? Glucose. 1, 2, 3, 4, 5, 6 into a 3-carbon sugar, right? So essentially, we're going to split this in half, and we're making 2 3-carbon sugars, which are essentially called pyruvate, right? And these 3-carbon sugars called pyruvate are put into phosphorylated oxidation and made into 2-carbon sugars, right? And these 2-carbon sugars are used up during the citric acid cycle. So you can think of every time you lose a carbon, you're going to release CO2, right? So here, glycolysis, we're breaking these 6 into 3. So we still have 6, right? We have 3 plus 3. But in pyruvate oxidation, we're removing 1 per pyruvate, right? So we're releasing 1 CO2, if you remember, over here. In the citric acid cycle, we're using both of our carbons left. So essentially, we release 2 CO2s. But that's just like a little extra information. Our question is asking G3P. So it's a 3-carbon sugar. Well, the only place we know that we have a 3-carbon sugar is during glycolysis and the start of pyruvate oxidation. And we know that a 3-carbon sugar in pyruvate oxidation is called pyruvate, right? So in this case, we have G3P. So this G3P needs to get modified into pyruvate somehow, and that can only happen in glycolysis, right? Because that's where we make pyruvate. So the answer to this question would be C, glycolysis. So number 5 is saying that we have a protein, bacterial rhodopsin, that's found in the plasma membrane of bacteria. So remember, bacteria don't have any organelles, membrane-bound organelles. So we're just going to be dealing with the plasma membrane and the cytoplasm. It can absorb light energy, okay, which causes the change in shape of a protein so that a proton is moved from the inside of the cell to outside using a channel protein, I'm guessing, right? And after the proton has been moved to the outside, the protein goes back to its previous shape, and the protons can then flow back into the cytosol through an ATP synthase. So it's looking like this is pretty much what we're seeing during the oxidative phosphorylation stage of cellular respiration, right? Except it's occurring in bacterial form. So instead of mitochondria, just think of it as a bacteria cell, right? Now, it's asking what type of process is this, right? Well, let's go through each one. Exocytosis is taking something in a vesicle or secreting something in general outside of the cell. Well, in this case, we are secreting protons, but they're going through a channel, number 1, and number 2, we're also taking them back in and producing ATP. So it doesn't exactly define what we're doing, right? So A cannot really be it. B, passive transport. Well, passive transport would be saying that we sort of went through diffusion, right? And the thing is, we're using this light energy, right? If you look at right here, it's saying that a light energy kind of instigated this change. Similarly, in cellular respiration, when you're oxidizing those NADH carriers to NAD+, you're using those electrons as energy to put your protons out. So same situation in this case, you're actually putting in energy, so passive transport won't really work. C, phosphorylation. Well, phosphorylation is occurring to make ATP, but that won't really define the entire process here. So it's not exactly what we're doing. And D, active transport. Yeah, so definitely active transport is what's going on in this case. We're taking our protons from the inside and using light energy to put them out of the cell, right? And then they're coming back in through a proton motive force through ATP synthase. So yeah, definitely active transport in this case. And then E, penocytosis. Penocytosis is taking in like water or small ions into the cell. It's a form of endocytosis. So it's something that we're not really covering in this case. So yeah, D in this case. So 6 is asking us about cellular enzymes in chemical reaction. So let's look at the answer choices. A is saying use energy made from ATP hydrolysis to force the reaction to occur. That's not really the case all the time. It's too much of a specific enzyme. So I don't think A would be a right answer in this case. B, decrease the amount of kinetic energy that's present in the system. Cellular enzymes don't really have much to do with the kinetic energy. They're more of the activation energy, right? So B doesn't really make sense. Same with C. We can take that one out either. Same. Reduce the energy needed for substrates to reach the transition state. Well, this sort of makes sense, right? So let's draw a normal energy diagram of a reaction. So we have our reactants, and then we have our transition state, and then we have our products, right? So it's products, reactants. This right here is our transition state, right? And this is our activation energy. And this, just for review, is our delta G, okay? Anyways, it's saying that it reduces the energy needed for substrates to reach that transition state, which is what we have seen throughout the course, right? We see that this would be a normal reaction without an enzyme. And if you add an enzyme, it would essentially sort of be like this, right? So you're reducing the activation energy needed in order to reach that transition state so your reaction can occur easily, right? And so D definitely sounds like something that's occurring in this case. So let's mark D for now. E is saying lower the overall change in free energy between reactants and products. So the number one rule when you talk about enzymes is that they don't change the delta G, okay? So delta G is not affected at all. Because if you look at this graph right here, delta G is down here. Enzymes only affect the activation energy, right? So just remember that delta G stays the same no matter if you add enzymes or not, okay? So in this case, they're changing activation energy, so D. So number 7 is asking us to compare alcohol fermentation versus lactic acid fermentation. A big thing to remember, just for review, is that this occurs in the cytosol, and it occurs when oxygen is not really present, right? So let's look at it in this case. What's the difference between the two? Well, you can see that you're getting pyruvate, right, over here. Both of these start with pyruvate because glycolysis occurs. And then you break that down to regenerate NAD plus from NADH, right? And in lactate, you can see that you still have a 3-carbon chain. So pyruvate has 3 carbons, 1, 2, and 3. And lactate has 3 carbons, 1, 2, and 3. While on alcoholic fermentation, pyruvate has 3, right? However, you're making a 2-carbon ethanol, 1, 2. So we lost a carbon, right? And what did we say carbons are lost through? Well, in the form of carbon dioxide, right? So that's why the difference, major difference between alcoholic and lactic acid fermentation is that alcohol fermentation releases carbon dioxide, all right? So let's take that into account when we're answering this question. So it says, if we compare both, so A is saying both break pyruvate to make a 2-carbon molecule. Well, we know lactic acid actually has a 3 carbons, right? So that's not right. That's only alcohol fermentation. B is saying alcohol fermentation produces less ATP. Nowhere in this case do we see any ATP production. In fact, we're recycling the NAD plus carriers. So in less ATP. So basically ATP is not a question in this case, all right? Only alcohol fermentation produces carbon dioxide. Well, yeah, we know that's a case. Alcohol fermentation, we see that we have CO2 over here, while lactic acid only makes while lactic acid fermentation only makes lactate. So yeah, let's keep C. D says only lactic acid fermentation oxidizes NADH to make NAD+. They're both doing that process right here, right? They're regenerating NAD+, so D is not right. And finally, E, LF requires oxygen, but AF does not. That's not true. Both of them occur under no oxygen, right? So yeah, C is our final answer. Okay, so number 8 is saying we have a biological sample containing a mixture of animal and bacterial cells, all right? So remember prokaryotes and eukaryotes, and it's viewed under a microscope. Structures shown on the right are seen. Of the following, which of the structures have come from? So let's go through each one. So A is saying an animal cell cytosol. That we can go over that. So let's look at our options. We have animal cell cytosol, bacterial cell cytosol, animal nucleus or cytosol, or either bacteria or animal, right? So let's take into account to figure out whether we're dealing with bacteria or animals or both first, right? So let's look at this picture. Well, what's happening is we see that we have a DNA strand and it's being transcribed into RNA, right? So we're seeing some transcription. So I'll write that down. I apologize for my handwriting. And then we're seeing a ribosome make RNA into a protein while it's being transcribed as well, right? So we're seeing translation and transcription happening together at the same time, right? Well, what do we know about animal cells? Animal cells have a nucleus, right, where a DNA is transcribed to RNA, which has to come outside of the nucleus in order to be transcribed or translated by a ribosome, right? Well, in this case, everything is happening in one spot. So we know that's for sure not possible in an animal cell because we have membrane-bound organelles. So you cannot translate something in the nucleus as you're transcribing it. So essentially, anything with animal cell in this answer will get canceled out. So E, D, C, and A are all wrong. B is your answer. And to further Explain why. Well, bacteria cells don't have any organelles, right? So they just have one plasma membrane and everything was over here. So their DNA is over here and they also have ribosomes in the same spot. So say that you start transcription. So I'm gonna draw that in blue. So your red is your DNA and you start transcribing this mRNA. Well, this ribosome is going to detect the mRNA and want to latch on and just start translating, right? So you can just have a ribosome latch onto this end and start translating it as it's still being transcribed. So this can only happen in a bacteria because there's no membrane-bound organelles, all right? So the answer is B. All right, so number 9 is dealing with familial hypercholesterolemia, which is one of the diseases that you guys talked about in your lectures. And so what is it? Well, hypercholesterolemia. So hyper means too much of something, right? So just in this case, we're talking about cholesterol. And in our body, cholesterol is transported through something called LDL, all right? Low density lipoprotein, right? Well, what happens is this travels in your blood. So there's a lot of LDL in your blood and it needs to be taken into your cell, right? You need to get the cholesterol out of your blood and put into your cell so you don't clog up your arteries, right? So what happens is you have receptor-mediated endocytosis normally. So you'd have right here, these are LDL receptors, all right? And let's just say this star is our LDL. So LDLR. So what happens is when LDL comes and it's going through a bloodstream, it binds to a receptor on a cell saying, hey, LDL, I'm LDL, and I want to enter your cell. So you'll have a bunch of LDL bind to these LDL receptors, and it will get taken into the cell by endocytosis, as we see here, right? So you're essentially getting rid of it from the bloodstream. Now, what happens with people that have hypercholesterolemia? Well, what happens is this LDL receptor is actually mutated, right? And it doesn't work. So if it doesn't work, then LDL cannot bind to it correctly and it won't get taken in. So what you'll have is a bunch of LDL in your blood, and this eventually leads to the clogging of arteries, and it's very dangerous, right? So let's go through each problem and see what answer makes the most sense. So family hypercholesterolemia is a result of, so it's asking why it's occurring. And A is saying the liver is making too much cholesterol. That's not really the case here. We're actually just lacking something of the receptor is not functioning right. So that's probably not the case in this case. B is saying a diet that is too high in animal fat. We don't know that correlation with receptors. So receptors more of like a genetic thing, right? Because you transcribe and translate these proteins, which eventually become receptors. So B is not right. C, making too much of the LDL receptor. Well, if you have a lot of LDL receptors, that's actually a good thing, right? You're going to get rid of it from the bloodstream a lot faster. So that's actually the opposite, all right? D, a defect in a type of endocytosis. So this is probably one of the most correct answers we have in this case because it's a generalized answer. So we know that it's taken up in receptor-mediated endocytosis, right? So if there's a defect, it can be anywhere. It can be in the vesicle formation. But in this case, we know it's in the receptor formation, which kind of is encompassed in our answer. So D for now sounds right. And E is saying being unable to make cholesterol. Well, if you don't have cholesterol, you can't really break it down, right? So there's no point, which doesn't really make sense. So in our case, D is the answer, a defect in the type of endocytosis, which is receptor-mediated endocytosis. All right, so number 10 is asking, of the following, the best way to describe what an ATP synthase does is, so let's talk about substrate level phosphorylation first. Well, what is substrate level phosphorylation? Well, that's using like an enzyme and a substrate to make an ATP, right? So that we have seen in glycolysis, and we see it in the citric acid cycle as well. We don't see it in the ETC or chemiosmosis because that chemiosmosis is using a process called oxidative phosphorylation, right? So that's two different things, all right? So A is not right. ATP synthase takes part in something called oxidative phosphorylation, all right? B, facilitated diffusion. Okay, so this can possibly be an answer that we'll review in a bit, but let's look at our other choices too. We have C, NADH oxidation. So this occurs in the electron transfer chain, but it doesn't have anything to do with ATP synthase, right? We're just using what happens in NADH oxidation. We're using those protons to help us out, but it's not a direct correlation. So C won't be the answer in this case, and D, make a proton gradient. Well, what makes a proton gradient is the electron transport chain. What uses the proton gradient is ATP synthase. So D is not right either. So that leaves us with B, but let's talk about why. So say this is our inner membrane, right? And this is the intermembrane space, and this is the matrix, right? And we have a bunch of protons going out against their gradient, right? Through the electron transport chain. So we have a bunch of H's, all right? I'm just gonna write a couple H's here. And over here, we have our ATP synthase. So synthase, all right? And what's happening is we have a high concentration of H, and we have a low concentration of H in the matrix, right? So we have a high concentration of H in the intermembrane space and a low concentration of H in the matrix. So generally, we know that if something has high to low, you're gonna go through a process called diffusion, right? Because it wants to even it out. So this, the protons here have no problem coming through this protein channel in order to even the amount of protons on either side of the membrane, right? No energy is needed. So it's gonna go through facilitated diffusion. Now, what's facilitating it? Well, it's this ATP synthase, right? Because protons are charged, so they're not just gonna travel through the membrane on their own. They need something to facilitate them. And that's what this ATP synthase does. So in this case, B is our right answer. It uses this gradient that these protons provide when they go through the protein to make ATP, right? So yeah, 10 is B. All right, so 11. 11 kind of encapsulates a lot of topic 6, I believe. And we're talking about enzymes and basically thermodynamics in this case specifically. So synthesis of a storage polysaccharide from glucose monomers is considered to be a blank. So it's saying we're going from a bunch of monomers. So say this is our monomers, and we're making a polymer, right? So in this case, monomers of glucose are called monosaccharides, and a polymer is called a polysaccharide, right? That's from last midterm. So we have our polysaccharide right here. And basically what we did was we made some bonds, right? And it's called an anabolic reaction, right? Because in an anabolic reaction, you're making something, right? So you're building up molecules. While a catabolic reaction, you're breaking down molecules, all right? Keep that in mind. So if you think about glycolysis, you're actually doing a catabolic process, right? Because you're breaking down glucose to pyruvate. You're taking it from 6 carbons to 3 carbons. While anabolic is a perfect example right here. You're making polysaccharide from a monosaccharide, right? So E is our possible answer. We know that B is for sure not right. Now let's talk about thermodynamics, all right? Well, when you have to make bonds, right, that's going to take energy, right? Because these bonds can't really form on their own. However, when you break bonds, it releases energy, right? Because you have energy stored in bonds. So a more clear example would probably be ATP. So you see we have adenine triphosphate, right? It has 3 phosphates. And if you remember, the reaction is you break ATP down to ADP plus PI. So you're breaking this into ADP, which is 2 phosphates plus 1 phosphate on its own. So since you broke off this bond, it released energy. So this is a catabolic process. So catabolic processes usually release energy. If you're releasing energy, you're most likely going to be spontaneous because you have a negative delta G, and that makes you an exergonic reaction, right? However, so that just knocked off all our answer choices A, B, C, and D. When we're talking about synthesis of bonds, so we're making bonds, it's going to take in energy. It's going to have a positive delta G most likely, and it's going to be endergonic, right? So in our case, our answer that makes most sense is E, anabolic, right? And also you can just do a series of elimination. You know that exergonic, negative G, and spontaneous are all the same thing, right? Essentially, they all fall into the same category. So A, C, and D can automatically be knocked out in this case, and then you'd have to choose between B and E. And you know that catabolism is breaking down, while anabolism is forming, right? So in our case, it's E. All right, so here we're talking about balancing osmolarity, right? So it's saying a beaker of 1 liter of water has zero point 0.1 moles of NaCl dissolved to it, right? So the water here has NaCl, all right? Two small artificial cells that are added are surrounded by a semi-permeable membrane. So semi-permeable in this case means only permeable to water, right? So no solutes, right? Cell 1 contains 0.1 mole of glucose. So I'm gonna make the cell 1 and it has 0.1 moles of glucose, while cell 2, which is right here, has 0.2 moles of glucose, right? So 0.2, all right? What happens to the two cells over time? Well, let's see what happens, right? So we know that we have these two cells, so I'm just gonna draw out this beaker over here just to make it a lot larger. We know that these two cells are dissolved in 0.1 molar NaCl. Well, let's calculate how much molar we actually have, right? So NaCl is held by an ionic bond, right? So you can break those into two ions, Na plus and Cl minus. So essentially, if you have 0.1 of NaCl, right? So one thing, and you know that these break down in water. Since you're putting in water, which you see right here, it's going to break down to two things. So we're going to have 0.1 amount of Na, and we're going to have 0.1 amount of Cl, right? So essentially, this 0.1 is actually going to be 0.2 molar in water, right? Because we're breaking it down into ions, right? So let's say we have, so on the outside solution, we have 0.2, which is what I'm going to put here, right? Now let's see what's going to happen. Well, so our first cell, which is cell 1, has an internal concentration of 0.1, right? While the outside of the cell is 0.2. Well, what direction does water like to move? It likes to go to the high concentration so it can even it out, right? So what's essentially going to happen is water from inside cell 1 is just going to leave, right? And this cell would shrink and lyse, right? Or not lyse, but it just shrinks, right? Because the internal concentration is smaller than the external concentration. In this case, cell 2, we see that both of them are at 0.2 molar, so they're both isotonic in this case. So it's all going to be the same, right? So in this case, we know that one shrinks and 2 stays the same size. So our answer to number 12 is A. Just to go over a third effect, let's say I had a cell that had 0.3 molar glucose inside of it, right? What would happen? Well, since you have a higher concentration inside the cell than outside the cell, this water will push into the cell and make the cell hypotonic, which may burst it if it's an animal cell, right? So yeah, that's just like the three possibilities. In this case, our answer choice is A. All right, so this is our last question of the day for the video. It's talking about phospholipids interacting with the oil droplets surrounded by water. All right, so this gray area is the oil, and imagine that this whole white area is a bunch of water, right? So let's talk about the structure of a phospholipid. Well, we know it has a phosphate head and a hydrophobic tail, right? Well, essentially, what does that mean? Well, if you have a phosphate head, it's going to be hydrophilic, right? It's going to be polar, so it's going to interact with water, right? So I'm going to put H2O liking. While a hydrocarbon chain is nonpolar, right? So nonpolar does not like water, right? It would want something more nonpolar, just like itself, so something like oil, right? So in this case, let's see what happens. A, we see that the heads are interacting with the oil, right? Well, we know that polar plus nonpolar does not really go along, right? That's not good. We want either polar to polar, or we want nonpolar to nonpolar, all right? So let's try to match these criteria up for each of these, right? So A, we know is not right because we're seeing a polar with a nonpolar mix, right? So no A. B, you're kind of splitting in the middle, right? You have a half of a polar head touching the oil and half of the hydrophobic tail touching the water. You're still violating the rules, right? You're having polar mixing with nonpolar, which is probably not how it's going to interact, right? C, you see that there's an entire phospholipid in the oil. So you see that as soon as you see a tail in the water or a head in the oil, you know that you're violating this rule because you're in this scenario. So C is not right either. D looks like something we would like to see, right? Outside is oil. We said that the heads like to interact with water. I mean, outside is water, my bad. And we said that the heads like to interact with water. So we're meeting the polar to polar check. Inside are the tails and inside is the oil, right? So we're meeting the non-polar check. So D looks like a good answer. E, we're seeing that we have a head interacting with the oil, and on the outside we have the tails interacting with the water. Certainly we know that's not it, so E is not right. So in our case, our answer is D. Other than that, that wraps up the first half of this sample midterm. I'll be going over the second half for Wednesday's video or Thursday's video. And yeah, study hard and good luck on Friday.